I saw a thing that kind of compared with the little squid of.
What is it?
Yeah, the squid of lemon.
Oh, interesting.
Also, it didn't really change.
All right. So, welcome to topic 5, where it's all about CRISPR. Um, and it's not that bad of a topic. It's just an overview. I think if you guys made it through VDJ, this should be pretty easy. So, all right. So, my computer didn't like that. Okay. So, a quick outline of what we're going to talk about today is first, just like you don't really need to know deep into the mechanisms of double-strand breaks and the repair in eukaryotes, but they are relevant. So, we're just going to go over them real quick. So, that involves HDR and HEJ. And then the history of genome editing as far as like what led up to CRISPR. So we had like ZFNs being used, TALENs being used. And we'll like talk about the differences of those. And then the mechanism of CRISPR and how it exactly works and the parts that are used in it. Gene knock-in and not out, like it's used for the CRISPR is used for gene knock-in and out. And then some controversy talk about who's credited for discovery and the patent disputes and stuff like that, because those are pretty relevant and kind of unique in this topic, because we didn't really get to see a lot of that in the others. And then just like a quick discussion on applications of CRISPR and how it can be used other than like just simple genome editing, because it's pretty versatile. Okay, great. I'm gonna close that door because it's a lot of them.
Trying to like mimic each other.
All right, sweet. So we'll first start with homology directed repair. So you probably saw this in 285, right? This is the most, so the most common form of homology directed repair, because it is kind of an umbrella term, is homologous recombination. And generally, homology directed, as in the name, can only be used when a homologous piece of DNA is present, because it needs that template to build off of and like reinsert the DNA, right? That it needs that template to do the complementary base pairing and fix whatever double strand break exists. So like I have questions riddled through here, so it's going to all of you guys. But what would so like when would this occur? I think Laura talked about this really quickly in class. Like when would we see HDR? Like what stage of the cell cycle maybe? Not so much stem cells, but think like generally cells, like when would a homologous piece of DNA? Yeah, so when it's replicating, right? Because that's when you're gonna have that template of like a template of where the double strand break would be in like that other chromosome, right? In normal cells with a simple genome, there's no other way you're gonna get that template unless it's inserted externally through like some, experimental method. Just in normal cells, the cells are only gonna revert to HDR when there's that template. So that happens when replication is going on, right? S phase. Yeah, and so once a double strand break is detected, proteins will come in and introduce this like 3' single-stranded DNA overhang. And then it uses that homologous template as a guide, and then the normal cell machinery, like the polymerases and all the complexes of proteins, will just finish off from that overhang over based on that homologous template. This is great because it's compared to non-homologous end joining, which we'll talk about in a sec. It's way less error prone because you have an actual template you're building off of, so it's kind of hard to go wrong. But it's one, rare, and two, it's like having that DNA template is unique to the replication phase, and it's just not feasible to do as easily maybe in other cell stages, right? And then we have non-homologous end joining. So this is kind of like the other end of the repair. So this compared to HDR doesn't require a homologous template. What happens is we have these proteins, Ku proteins they're called, and this complex binds the double strand break first and prevents the degradation of the ends, because that's important, right? You don't want to degrade more DNA and just have this bigger break. And then it recruits the non-homologous end joining machinery. And depending on the gap, like the size that's present at the double strand break, if it's small enough, this can go through simple ligation where DNA ligase, IVXR-CC4, don't need to know that. This complex will just like complete that ligation and stitch them back together. And if it's larger, you're gonna recruit the DNA polymerase to fill in the gaps until that's supposed to say ligation occurs. Now, the issue with this is that both these methods can introduce a gain or loss of nucleotides because the non-homologous end joining process will kind of, it doesn't have a homologous template, remember, so it's going to kind of just like randomly insert nucleotides or kind of almost guess as to what should go there, right? And this is obviously way more error prone because you're not ensuring that you're getting the right base pairs in there, but it's quick, it's efficient, it stabilizes the DNA, prevents the cell from going into apoptosis. And it's not restricted to S phase or replication stages of the cell cycle, right? Because you really only need, that's fine. You really don't need that homologous template present to just do this. And so cells will generally, the majority, like anytime a double strand break happens in like a normal situation, tend to go to this route with the idea of maintaining the DNA, preventing further degradation of the genome. Yeah, we'll talk about this in a sec too, but the use of like because the CRISPR's whole process is kind of introducing a double strand break, and then depending on what your intention of outcome is as far as like, are you knocking in a gene, are you knocking out a gene, that will change what sort of repair mechanism you're going to shoot for. And I'm sure you can see that already. Like if you're looking for, we'll talk about this in a sec too, but like if you're looking to knock out a gene, for example, using CRISPR, you're gonna probably go for non-homologous end joining because you want those random insertions. You want that gain or loss because that's what's gonna make the loss of function in your knockout happen. Right. So stuff like that is what's considered when you're thinking post double strand break in CRISPR. Any questions?
So to like give in the template, like you introduce the CRISPR mechanism, whatever.
Mhm.
And then if you want homologous end joining, can you also add that template?
I think so. Yeah, as long as you introduce the template at some point, and there are studies that do that, then you can go through homology directed repair and kind of use that machinery to. Now, I'm sure that's harder than doing this method, right? But I think it is possible. Okay, great. And then, so as far as use for up to CRISPR, there were a couple things that were used back starting in 1996. And the first was the zinc finger nucleases or ZFNs. And this is one of the first genome editing tools. It consisted of a DNA binding domain and a nuclease domain. So the DNA binding domain were made up of these zinc finger proteins, and a nuclease domain, which actually did like the cleaving, was an enzyme called I don't know how to pronounce FOKL or like focal. I don't know. That requires, and this that enzyme requires dimerization, so they need to come together, and then the cleavage occurs. Now the great part of it is to describe like a general concept, if it worked, but it wasn't perfect. And it was pretty difficult to design and expensive to just make because each ZFP or a zinc finger protein recognizes a specific DNA triplet. So you can see that in the image up there, right? You have 3 ZFNs per side, right? And then you have like each of them kind of recognizing a triplet of nucleotides. So think about it. If you're like trying to target a sequence that's, I don't know, like 20, 25 base pairs long, or I guess we can do a multiple of 3, right? 24 base pairs long. You're gonna have to design like 8 separate proteins that like have different properties to bind those nucleotides. It's not as simple as CRISPR, which we'll talk about in a second, making like an mRNA molecule that can be served as like because mRNA is easy to make, but designing proteins like this is kind of hard. And yeah, and that small number of bases in the binding region and those like just identifying triplets per triplet kind of introduces the potential for off-target effects, right? Because if you're going by triplet, chances are there's similar areas in other proteins, and we'll talk about this in a sec too. But CRISPR has that PAM sequence, which kind of like is like a double. It's kind of like, okay, you have the target sequence affinity, but now you have the PAM sequence too, which will like ensure that we're at the right location, which this doesn't have. So off-target effects were kind of a problem with this. And then that leads us to TALENs, or transcription activator like effector nucleases. So this consists of a tail DNA binding domains in the same enzyme, the FOKL nuclease domain. So tail stands for transcription activator like effectors. And these are able to recognize specific DNA sequences as well. Now, the difference was that these tails can recognize one base pair. So it's not triplets anymore, but now we can go to one base pair. And this is great because it improves specificity over the ZFNs, right? Instead of working in triplets, now you're working base to base. But similar to the ZFPs, the production of the TALENs is pretty difficult still and costly, right? Now you're designing proteins for every base pair. Yes, your specificity is better, not perfect yet, because we still do have that idea of off-target effects being apparent because, you know, you don't have that PAM sequence and whatever. But you still have to design each of these proteins and each of the base pairing interactions. So it's just it's like a drag almost, for lack of better words, right?
Be careful with this next one.
Yeah, I would think so. I think, yeah, like I think this would be more specific, and it is more specific, but I could see it being more expensive and costly to do. Yeah, in the same sequence, the same like structure. You just have those proteins that bind the sequences and then cleavage in the middle. And don't quote me on this because I'm not exactly sure, but I think the cleavage wasn't because it's like you're binding around the region and then cleaving in the middle. You're not exactly 100% sure you're cleaving at the right spot, which doesn't happen in CRISPR. With CRISPR, the cleavage is like down to the base pair, correct?
So here you're not binding to the site you're cleaving. You're just binding to the site.
I believe so. Yeah, the way you're cleaving it is in the middle, and that's going to be like you're almost surrounding that site. Um, yeah. Okay. So this leads us to CRISPR, right? Basically, CRISPR is a natural defense mechanism in bacteria against viruses, which was discovered, and we'll talk about this in a sec too, of who exactly discovered what, but it was present in bacteria before anybody even designed synthetic versions or anything like that. So once a virus is introduced into the organism, parts of it are incorporated into the genome as a spacer sequence. So it's not the whole virus genome, right? Just like parts of it. And the spacer is surrounded by CRISPR repeats and spacers, and other spacers, and this is called like the CRISPR array. Right? And from this CRISPR array, a pre-cRNA molecule is transcribed. So that's that big long stretch over there where it says pre-cRNA with those like little loops. And then it's a Cas protein is used to kind of split this up into smaller spacer sequences, which are the cRNAs or mature cRNAs. And basically, this serves as a form of immune memory because what you're doing is you're taking the virus, parts of the virus genome, integrating it into your DNA, putting those spacers so it's not targeted after you have like the production of the mature cRNA. Those spacers are important so you don't like break up your own genome. And then producing cRNA that will just like kind of float around and like prevent or work against like repeated infection. Right. When reinfection occurs, the Cas proteins bind and disable the virus through enzymatic activity. And those spacers, like I said, are important because the Cas proteins don't bind your own genome. They only bind the virus once it's reintroduced. Does that make sense? Cool. Okay, so that leads us to like the parts of CRISPR that are used for our purposes. So we have the Cas proteins. Many Cas proteins exist. We're just going to talk about Cas9, but I'll touch on the other ones. We have a guide RNA or a gRNA. This is made up of CRISPR RNA, cRNA, and a transactivating CRISPR RNA or a tracer RNA is what it's called. And then you have your protospacer adjacent motif, so your PAM. And we'll go through each of these individually. First, I guess the star of the show, right? Cas9. This is great as far as cost and efficiency goes because you can just harvest it from bacteria, right? And that makes it like you don't have to design these like proteins that do the cleavage like the ZFNs and the TALENs. You can just harvest it from bacteria. And instead, which we'll talk about in a sec, introduce a mRNA that will like serve as a targeting mechanism. But basically your Cas9 protein has two recognition domains. You have a target recognition domain, or a TRD, and a protospacer adjacent motif, PAM, interacting domain. So both of these domains are important because one, the TRD serves for the target recognition, right? You're finding that target DNA in the genome. And then the protospacer adjacent motif, or the PAM, will also find that PAM sequence in the complementary strand to the target strand, right, on the other side, and ensure that we're cleaving and working in the right spot. So the Cas9 also has nuclease domains, and this is what actually does the cleavage. It's called an HNH. Is the actual like domain name. I don't think you really need to know that, but it's there. And so the whole Cas9 protein is kind of loaded with the guide RNA that directs it toward the intended cleavage site. And like I said, PAM must also be present for cleavage to occur. And then once these two conditions are satisfied, the bound Cas9, so once you're like bound to the target DNA and you have that PAM there, it undergoes a conformational shift that activates the nuclease domain. So the nuclease domain only happens when you have both those things there, and both those things are interacting. Any questions on Cas? Great. Okay, and then guide RNA. So this is a synthetic RNA molecule made up of two parts. Again, cheap and easy to produce. mRNA is way easier to produce than producing the actual like proteins, like the ZFNs or the TALENs. So it's made up of CRISPR RNA, cRNA, and this is complementary to the target DNA sequence, and typically around 20 nucleotides in length. And this is kind of gonna like, how do I show this? This is like this part right here. It kind of runs up here, right? And then because you also have your tracer RNA, and this is used to link the cRNA to the Cas9 protein. So I kind of like to think of this as like the anchor to the protein rather than like finding the targeting. And this has a stem loop structure that is complementary to part of the cRNA. So, I mean, it's not perfect here, but you can imagine the cRNA goes like here, and then you have complementary base pairing with the tracer RNA here, and that's what kind of like cements it into the Cas9 protein. And yeah, it also contains that region that helps recruitment of the protein. And so I like to think of it, like I said, tracer kind of interacts with the Cas9, puts the target, the guide DNA, guide RNA strand there, and then the CRISPR RNA, cRNA will be the actual one that's like complementary binding to the DNA sequence that we're targeting. Cool. Any questions? Sorry, I'm like in your way. Okay. And then we also have that PAM sequence. So this is a short sequence of 2 to 6 nucleotides that is highly conserved. Different Cas9 proteins can bind different PAMs. This adds to increased specificity of CRISPR, right? Because not only are you now like specifying by the target strand, but you can specify by the PAM sequence present there too to see if you want to actually go through with the cleavage. Generally, the form of the Cas9 one is 5' is NGG to 3', and N just stands for any nucleotide. So you can have like any nucleotide there, and then a guanine. And this plays a role in recognition of the Cas9 system to the target DNA. But it also creates a limitation in the application of CRISPR with specifically Cas9. So I don't know if you guys have any ideas like how could like you potentially overcome, like let's say you want to target a strand of DNA in the genome, but it doesn't have the correct PAM, right? You have a different PAM that that's there, and your Cas9 obviously isn't going to work there because the PAM doesn't line up. What sort of options do you think exist that can overcome that limitation? It's cool if you don't know. I didn't know before I did this slide. I wouldn't break. See, I wouldn't break the PAM. No, and you can't do that either. You don't want to touch the PAM. But what you can change is specifically the Cas you use, right? We're talking only about Cas9. Cas9 is like the most widely available and easy to harvest. But other Cases exist, and those other Cases have different PAM binding affinities in different regions that they can bind. Like this Cas12 will do like a TTN or a TTTN, right? And if that's something like that's surrounding your target DNA, then you might want to look into options using Cas12. Now, this is something that's like still being researched. Cas9 is like just like the most widely used, and different Cases are still being discovered. But basically, what's conserved in your Cas9 use across any DNA that you're targeting is that PAM sequence. So if that PAM sequence doesn't line up, you have to looking into different Cases, because that's what's conserved across all Cases. You can't change, like, you can change the target or the CRR RNA in any application you use, but you can't use that. You can't change that PAM affinity. You have to use different Cases to achieve that. So you use a different Cas, but use the same maybe like target DNA to hit different points
The PAM is always in the virus, right?
in the bacteria virus in a phage. Yeah, I think like, yeah, it will be. Yeah. Okay, cool. Yeah, sure. Yeah, we have PAM sites all over our genome, which can be used. Doesn't necessarily mean it's at the specific point you want to target, but they are present, and um, they can be like found, you know. Yeah, so that's why Cas9 is in most applications able to be used, but doesn't mean you can immediately target everywhere, is what I'm trying to get at.
Well, if it's that RNA has to be really good.
Or that, you know, because, yeah, so you're asking like why he doesn't bind to all different sites? Is that your question? Sure. It's the target. Because it might go there, but but a majority of the affinity and where it's gonna be directed to is based on the CR CRRNA, right? So that target still has to line up. And the fact, like, the chances of your target, we can go back here, your CRISPR RNA, your CRRNA lining up, which is 20 nucleotides, and the PAM lining up is pretty rare for it to happen in two spaces. It still can, and we still do see off-target effects. And that's kind of why, like, research is still being done. We don't have like a wide application throughout the entire body, use in like just like immunotherapy and stuff. But like the chances of it hitting two spots is rare, and that's why the specificity is way better than this like ZFNs or TALENs, because you have this like double check. Yeah, and remember the ZFNs and TALENs, while they can be 20 nucleotides in length, designing that is a pain, and this is really easy to do. Yeah. Any other questions? Okay, great. Okay, so post double strand break, the repair mechanism used after double strand break is introduced partly depends on like one, the presence of homologous DNA, because that might be HDR, and the stage of the cell cycle, because that might also define if you can use HDR or not. I kind of talked about this already, but for a knockout where the gene is turned non-functional, which repair mechanism would generally be preferred? And we said non-homologous end joining, right? Because that's what's gonna like introduce those insertions, deletions, which make a gene hopefully non-functional, right? You're gonna get a frame shift or a deletion or something that's gonna mess up that transcription of the protein. HDR would be preferred. I think if you're like trying to insert a specific genome sequence into that region where you created the double strand break. So you kind of like provide it the template, make the double strand break, and then the machinery takes over and reinserts that template or the complementary base pairing of that template into the genome.
Sickle cell, it's only one base pair.
Yeah. Yeah. Just on theory, I can assume that that's harder to do than non-homologous end joining, just like knocking out genes. Because knocking out, you're just like, all right, take over, do whatever, and just mess it up. Where over that, you have to like ensure that you're one, hitting the right spot, and then two, doing the right replication and inserting it back at the right spot. So it's just like it can be done, it's just, I guess, in my mind, it's harder. So I guess it's less utilized. Okay, and then the timeline of discovery for like that talked about in the papers. So starting from 1996, many advancements were made in the field of genome editing, right? ZFNs, talons, discuss these. And then up in 2020, 2012, Charpentier, I think is the way to pronounce it, and Dudna, discovered the CRISPR-Cas system in S. thermophilus, which is a bacteria. In that same kind of graphic I showed, and the other one, that's actually their Nobel, that was from the Nobel paper that you guys hopefully read. And basically they talk about this system and how it's used in bacteria, and they do discuss and list potential uses and applications in eukaryotic cells in their work. But initially, their papers regarding their work was rejected by multiple journals. Now, the exact reason for this isn't exactly cemented and not known why. I'm sure, like Laura talked about in class, right? One, the novelty of the technology may not have been known, exactly how important this was and how applicable this was. Two, there's a lot of politics, science, and unfortunately, like sexism still exists too. So that could be like a factor that played into why they took so long for them to actually get this like groundbreaking work published. But they eventually did. And you know, they were recognized for their Nobel Prize in 2020, but that didn't come before Feng Zhang and others at the Broad group and institute out in Cambridge. They successfully adapted their CRISPR-Cas9 for genome editing in eukaryotic cells in 2013, which isn't something that this Charpentier and Dudna exactly did. They worked within the bacteria, but listed uses for eukaryotic cells and like the kind of application that it can serve. But Feng Zhang was the actual one that did studies in eukaryotic cells and saw results. So this created a patent issue and a dispute, with the key issue being around Charpentier's and Dudna's work being in vitro, so like in the bacteria, and that being enough to claim patent rights of use in vivo or in actual cells, right? Was their work and the things they discovered in the foundation enough to claim rights over this technology's use in vivo? And some updates for that is the U.S. Patent and Trademark Office ruled that the Broad Institute's patents actually covered the use of CRISPR-Cas9 in eukaryotic cells in 2017. I know Charpentier and Dudna went through some appeals that I think got rejected, and other lawsuits were filed by them that are still ongoing. So this is like a still ongoing controversy and kind of you know, I want to say fight, but some clashing of heads, right? But you know, each of these groups are still doing their own research. I know specifically for like Feng Zhang did a lot of research for CRISPR's use in COVID, which we might discuss in a sec. So these groups are still working on everything, on specifically like CRISPR related stuff.
You said that to the Charpentier and
Yeah.
they did it in bacteria.
Yeah.
And then the other guy did it in.
Yeah, specifically what they found was that like the Cas9 system and the way it functioned in bacteria existed and it could be used to genome edit. They didn't do it in eukaryotic cells, right? That's what Feng Zhang did and their group did, but they found like the foundation for the machinery and how it worked. And that's, I think, like that's why they deserved and won the Nobel Prize for it in 2020. Yeah. I don't know if he thought for the Nobel Prize, but he definitely thought in their group was for the idea that they own the patent for use in eukaryotic cells, right? So like basically Charpentier and Dudna was like, okay, we discovered this mechanism and this thing. We want patent rights for use in vitro and in vivo, like any sort of application it might have, even though they didn't do the application. But they did discover like the foundation for it and the mechanisms behind it. And Feng Zhang was like, okay, well, we did the studies in eukaryotic cells and we kind of like did the research of how exactly we're going to use this in eukaryotic cells and how it works. And so we deserve the patent rights for in vivo use. And so there was disagreements about that.
You can just split it. into little things.
So the way I think about it, like the in vivo use patent is way more valuable, right? Because that's the only reason it's going to matter for us, right? Any sort of um, human cell interactions, like disease, like recognition, gene editing, disease, I don't know, introduction to mouse models, whatever, like those kind of things, like that's what's actually where the money is and where like most of the use is. So that's what's important. Yeah, it's kind of crappy on the Charpentier's and Doudna's end. But like I said, lawsuits are still going on, so I'm sure it's not totally totally resolved. And Laura knows a lot more. Like there's a lot of politics in the science game that I don't really know, that I'm sure Laura would love to talk. She's great at knowing that. So yeah, compared to the patent, that's like billions of dollars.
Anytime anybody uses it, you get money.
Yeah. Anytime somebody like uses it and licenses a product, yeah, you get a cut. Yeah, I would be too. That's a lot of money. Okay, great. And then last slide, I think, is just some uses of CRISPR that there are many uses of CRISPR, and not technically like all of them are in use right now, but like theorize and where we can apply it. Specifically like the ones we talk about, gene editing and therapy. So genome-based diseases can kind of be almost eradicated, right? If we can specify where a disease happens in the genome, go in, change it, fix it, days all better. Issues still exist as far as like specificity, because CRISPR isn't perfect. It's great. It's way better than anything we've had so far, but it's not perfect. Because of that, most trials right now are only being done in like closed body. I think Laura mentioned this, like closed areas of the body. So that includes like your eyes, right? Your eyes are in their own little system. So like I think CRISPR was kind of used to treat some form of blindness. There's still being research done into that. But also cancer. Cancer is interesting because there's so many different types of cancer. So who knows what type CRISPR is going to be most effective for, right? That image right there kind of shows that you can use CRISPR in this specific type of cancer to kind of change the DNA so you don't express that PD-1 anymore. PD-1 is important because PD-1, when it binds its ligand, it will inhibit if I'm remembering correctly, inhibit a immune response. So by removing that receptor, you are promoting a immune response against the cancer cells. So you can edit the genome to kind of just like, hey, we're not gonna produce PD-1 anymore. So these T cells now, the edited T cells will go ahead and produce the immune response against the cancer cells. Right? That's huge. Probably. Yeah. I mean, yeah, it's kind of revolutionized the way we go about kind of doing this, because before it was like a lot of considerations for drug therapy and mostly like drug delivery. But now if you can just like target the cells and kind of just introduce Cas9 into all these systems, and like with the guide RNA, obviously, and just change the DNA, then you can effectively just like clear cancer without any like dosage considerations or anything like that. It's cool. But CRISPR itself can also just be used simply for sequence identification, right? You can have the nucleus domain, which actually does the cleaving, to be altered to have a loss of function when you insert it. So what it'll do it'll do the same thing up until the point where it cleaves, where it'll like use the guide RNA, find that target sequence, find it, but then it's just like unable to cleave, so it's kind of stuck there. And you can kind of like put like a fluorescent maybe tag on the Cas9 protein, and you can say like, okay, let's see if this DNA is present there, and kind of find it really easily. And with, importantly, high specificity, right? Because you had that PAM and target DNA kind of like working together to make sure we're at the right location. That bottom figure kind of shows that there. You can use that for COVID. You can take like viral RNA from the COVID, amplify it, send in the Cas, and see if like specific strains of that COVID exist, right? Or if like COVID is actually present in that viral or that sample of DNA RNA, right? And then also animal models for disease. A lot of diseases, it's hard, like even before you start researching them, it's hard to induce them in the animal models that you're working with. Right? Like, I don't know if you're, give me a genome-based disease, anybody. Any genome-based disease. Sickle cell could be one, yeah. So like introducing sickle cell, I think like you could go into those mouse models, make the gene change to like promote that disease, and then that will let you like do whatever studying or whatever therapy trials you need to do, which is huge because that just speeds up like the first step or obstacle of research, right? Getting some models for use. Um, yeah, okay, sweet. That's all I had. Thank you for joining us, whoever joined us on YouTube. And I will stop this now. Oh,